All right, let's go ahead and get started. So, a couple of things. If you haven't taken a look at the announcements up on Blackboard, I posted one just to give some info on how the redos on the exam should be submitted and when the due date is. So take a look at those. I'll hand those back later. I'm going to lay it out up front today. This may not be the best lecture ever. I apologize. I'm kind of like all over the place mentally right now. So we'll do our best. We'll try to stay focused, but bear with me. And then 3, is anybody opposed to the flavors of Old Fashions, cake donuts, or the like non-cakey, the fluffy ones, whatever you call them? Anybody opposed to those types generally? Okay, that's what's getting ordered. So that's your incentive to come on Monday to actually work a little bit during class time. Okay, if you don't, no soup for you. Old Seinfeld reference. On Tuesday, you know you have nothing, no lab, no nothing going on. I'm going to be around all day Tuesday though, so if you have questions about your lab project, or if you have questions about the mini lecture that you're going to put together, I will be around, and so feel free to stop by. You don't need to make a meeting with me. I actually have nothing, and I'm looking forward to it. So I'm going to clean my office. That's what I'm going to do on Tuesday. So stop by. Bring a dish rag with you, and you can help clean it too. All right, let's start here. Where we finished off on Wednesday was talking about NK cells. I took a look at everybody's tables, and honestly, I'm not sure what you guys were frightened of. Those tables are really good that you had put together so far. I think that there's some modifications that can be made. I tried to throw some questions in there to get you to work on that, so take a look at those comments. And actually, I think these can be really good study tools for this final exam, particularly for these 2 objectives. Explaining the function of B, T helper, and T cytotoxic, and then comparing NK with T cytotoxic as well. So I want to start with 11.5. I want you to take 2 to 3 minutes, and I want you to compare both the structure, the activation, and the function of NK cells and TC cells, or those cytotoxic T cells. Focus on both similarities and differences. So maybe you create a Venn diagram to put this together, but I want you to take about 3-4 minutes to do that, and then we'll list out some of these characteristics. Cells. Should we draw this as a Venn diagram? Anybody opposed to that? Where shall we begin? TC cells, says Jamie. What do we want to talk about with TC cells? How do TC cells become, what are 2 molecules or receptors that we talked about are on the surface of TC cells? Ashley? The CD4 would actually be on the T helper cells, but CD8 is 1, and then Devin? MHC. The MHC is what they bind to. The TCR, the T cell receptor, is the other big one. I'm going to actually put, where would MHC go? MHC1. Right, it's on an infected cell. Is it something common between the 2 of them?
Well, one recognizes the presence of it.
Good. So there is a commonality in that they're both looking for MHC1, right? So MHC1 is a commonality, but in the case of NK cells, they are looking for the absence, which would indicate there's something not right about that cell. Whereas the T cells are looking for the presence with an epitope. loaded in that MHC1. One thing that we should point out here is the T cells, those TC cells are only going to respond to cells that have MHC1 with the specific epitope that those TC cells are able to identify. Okay. NK cells, it's basically any cell that doesn't have an MHC1, it's going to go ahead and kill. What kind of receptors do NK cells have on their surface? There's an inhibitor one and an activator one that we talked about, and we didn't really give them names, but there's an inhib activating receptors. Other commonalities or differences? Kevin.
Like the pathogens within the cell and TKs if it's on, like, before it gets into the cell?
In both of those cases, I think what you're trying to hit on is the difference between MHC1 and MHC2. So MHC1 typically presents intracellular pathogens, whereas MHC2 would present ones that have been phagocytized and brought into the cell. So that difference really comes down to what's the difference between TC cells and T helper cells. T helper recognize the MHC2, and particularly what we might consider to be external pathogens. Okay. These ones don't. No. What do they perform?
I was going to say lysis, like granzymes and perforin.
Yeah, that's a key point. So their response to the identification or lack of identification of MHC1 is the same. Both of them secrete perforin and granzymes. And we said that together these will lead to cell death and the end, hopefully, of that infection in that particular cell. One of the things that we didn't cover is which of these is an adaptive immune cell and which of these is an innate immune cell. And this would be a good opportunity to review what those two terms mean, or some contrast between those as well. So adaptive is oftentimes specific to a particular antigen or to a particular epitope. Which of these is specific to an epitope? Are TC. So TCs, B cells, and T helper are all part of the adaptive immune response.
What ones did you say that? PC?
T helper and B cells. And then on the other hand, our NK cells, macrophages, and neutrophils would all fall under the innate immune response, or part of innate immunity. Again, in innate immunity, one of the things we'll talk about is there's no memory. It's just it recognizes these generalized patterns of pathogens. Any questions on this? I think these are kind of the major points. I don't think we're missing anything based on what we've discussed so far. Okay. I suppose if you're looking for one last commonality, in part their activity or is enhanced or activated by T helpers. T helper cells and their secretion of cytokines are going to increase phagocytosis in macrophages and neutrophils. It will increase the cell-killing capabilities of TC cells and of NK cells as well. Any last questions on this, or anything else from what we've covered on Wednesday? Okay, let's move into B cells. Let's spend a little bit of time today talking about, one, how are B cells activated, and then two, what the heck is the role of a B cell? Oh, bugger. I'll only be able to do that, though, if I can find my mouse on my computer. All right, there we go. All right. The role of a B cell, produce antibody, produce boatloads of antibody. And antibodies are, sometimes the word you might see is immunoglobulin, is the other term that's used. Sometimes the abbreviation is IG. IG for immunoglobulin. And there are 5 different classes of immunoglobulin. I'm not asking you to know the different classes. But you may have heard terms like IgA, IgG, IgM, IgE, and IgD, which nobody really knows what IgD does. But each of them is involved in connecting the adaptive to the innate immune response. The way that they do so is somewhat similar. So in the structure of an antibody, we've got these ends here, which are just listed as these antigen binding sites. You can see that these are also the variable regions of antibody. And what we find actually is these areas undergo hypermutation. So we talked about, and I'm not wanting you to know this really, but I want to just kind of describe how do we get so much diversity. Our cells, B cells in particular, as B cells and T cells are being created to give us the diversity of T cell receptors and B cell receptors and ultimately antibodies, there is basically, it's encouraged in those genetic regions to undergo mutations. And sometimes those mutations don't produce an antibody, and those cells just end up getting killed. But those that do produce a functional antibody, they will have unique variable regions, but they'll all have the same conserved or constant region. And so the variable region allows for antibodies to recognize specific epitopes. Again, each one is each B cell produces an antibody that is uniquely designed to recognize a different epitope. In addition to this, the constant region, this is where what differentiates our IgA from IgM, etc. This constant region, or this tail here, can differ from one of those to the other. And the importance of that is it allows for these antibodies to interact with different immune cells. So for example, IgE pretty much interacts with, one of the cell types it interacts with are basophils, something we haven't talked about. But one of the roles of basophils is to secrete histamines. IgG will bind to macrophages. And we'll actually talk about the importance of that binding to macrophages to actually improve phagocytosis even more. IgA, on the other hand, primarily found in the mucosa of the human GI tract, in our nose, urogenital tract, mouth, etc. There is IgA coating basically every single surface. It's a neutralizing antibody. Its function is to prevent pathogens from being able to colonize on cells. This constant region is also known as the FC region. The reason why I bring this up is because we'll talk in a few minutes about what's known as an FC receptor, or an FCR. Okay, and the importance of that in binding to this region. So the main takeaway points, here's where an antigen binds. For each antibody, there are 2 binding sites. Those are the variable regions of the antibody, and then we have this conserved or constant region that is the same. And this, again, is how we connect the specificity of the adaptive immune response with the generalized response of innate immunity. Two types of antigen or professional antigen presenting cells. Anybody recall the two professional antigen presenting cells that we've talked about? The key characteristics are they perform phagocytosis and they can present pathogens on MHC2. Macrophages are one. Not neutrophils, actually. Neutrophils don't, they do perform phagocytosis, but they don't present the pathogens. Not as well. Cytokines would be the molecules that are secreted. What's the focus today? B cells. B cells are the other type. Macrophages and B cells are two of the major professional antigen presenting cells. And so in some ways, the initial steps of a B cell's activation is actually quite similar to what we find in macrophages. However, there's one key difference, and that's at the point of the attachment. How do macrophages attach to pathogens? What receptor do they have? PRR or toll-like receptor. Okay, and they attach to a PAM. B cells, on the other hand, in the early development of a B cell, their antibodies are actually membrane bound. And we call this the B cell receptor. So this B cell receptor being shown here will recognize a specific epitope of an antigen. In other words, the difference here in attachment is that for the B cell, it's specific to the antigen or to the pathogen, whereas for macrophages, it's generalized. Again, it's just based on a common molecular pattern. At this point, the B cell phagocytizes that pathogen. Phagosome fuses with lysosome, destruction of the microorganism, breakdown of the protein, and then presentation of all of those antigens on what MHC complex? 2, yeah, on 2. One of the things that I should probably note here is that for these B cells, this image doesn't do a great job of depicting it, but it can present multiple different antigens or epitopes from the same pathogen that it's just engulfed. And so that allows for it to interact with numerous T helper cells at the exact same time. Any of those T helper cells that recognize one of these epitopes that that B cell is presenting could allow for that B cell to become fully activated. The next step of this process, T helper cell docks. What is this interaction here? What's this T cell? This is the T cell receptor. Sorry, I just gave away the answer. But what else do we need to have? CD4 in this case, between the T helper and MHC2. So those are our two signals. T helper cell in response secretes cytokines. And in this case, the cytokines that it's secreting will encourage that B cell now to change gene expression so that all it is doing is producing antibody. At this point, 90% of the protein that is being produced in this B cell is antibody that will be secreted from that B cell. So it loads the antibodies into vesicles, releases them into the environment. Okay. So if you're thinking about that table that you've put together, a couple of the key characteristics of B cells, at least in structure, they've got the B cell receptor. They also will have MHC2 on their surface. Main function, produce antibody, and this is something that a lot of people noted. And they do phagocytosis as well, but that phagocytosis isn't at the same rate as what we observe in macrophages. It's not the main role of a B cell.
Also, in addition to producing antibodies, B cells start to replicate like mad at this point too. So it's kind of a way to increase the number of cells that are all producing the same exact antibody at this point. All right, what do antibodies do? What is the benefit of an antibody? If you were to look at our book, it would say there's 5 things that an antibody does. We're not going to go over each of those 5 things because, in all honesty, they basically revolve around the same exact theme. The job of an antibody is to bind to a pathogen or to bind to the epitope of a pathogen. And in binding, what we find is that can either improve phagocytosis, so that's one, inhibit the pathogen in some way, and by that, in some way I mean it could inhibit attachment. Sometimes the term that gets used is neutralize, because there are antibodies that will bind to toxins and neutralize those toxins and prevent them from binding to host cells and causing some sort of functional change. And the third thing is antibodies improve the innate immune system. So let's take a look at 2 of those here. On the left, what we're looking at is an image of staph epi. And what they're trying to model in this image is, here are some different surface proteins. Each one represents a different epitope in this case. A B cell has recognized this particular pathogen, engulfed it, has been stimulated by T helper cells to produce antibody. And now these antibodies, again, the variable regions here, those arms, are capable of binding to the epitopes on staph epi. There's a couple of effects that happen here. One, prevents staph epi from colonizing on the surface of host cells. 2, macrophages have Fc receptors, or FCR. And that is all of these little things down here. The FCR binds to the constant region or the Fc region of an antibody. What might happen as a result of this? We've bound a pathogen to that cell. What might happen next? Yeah, exactly. That's the role of a macrophage, is phagocytosis. So what we find here is that by coating that pathogen with antibody, this is another way by which macrophages can, in this case it's an indirect contact to the pathogen, but still increase the process of phagocytosis. This process here, where an antibody coats the pathogen, is called opsonization. O-P-S-O-N, opsonization. And that improves phagocytosis. So opsonization is the coating of a pathogen with antibody. And opsonization will increase the rate at which a pathogen is phagocytized and destroyed by a macrophage. Actually, we find these receptors on neutrophils as well. So both of those phagocytes. All right, I'm going to pause. See if there's any questions on what we've talked about in the past 5-10 minutes. I'll give you a minute or two to look through your notes. Any questions? Okay, let's let's take a look at one more example real quick, and then we'll come back to this image. But the example I want to do here is NK cells. NK cells actually also have FC receptors. When, let's say in this case, let's deal with a eukaryote. Let's say an amoeba, brain-eating amoeba. Yay! So brain-eating amoeba is opsonized with antibody that recognizes some surface protein on it. And those antibodies bind, the FC region of the antibody binds to the FCR on the NK cell. What do you think is going to happen? What does that NK cell do in return? Ooh. Kills them, right, secretes. So this triggers the same sort of response that the absence of the MHC1 does. The NK cell secretes perforin, secretes granzyme, and the amoeba dies. This is another process that antibodies are involved in. I'm not asking you to memorize this word, but if you want to attach a term to it, antibody-dependent cellular cytotoxicity, ADCC. And that refers to this process by which cells, eukaryotic pathogens, for example, are destroyed by NK cells from their secretion of granzymes and perforin after they have bound to an opsonized pathogen. Okay. So again, the takeaway point here is that antibodies tie together something specific and unique about the pathogen to the innate immune cells. And the antibodies are allowing the innate immune cells to just perform their normal function. Okay? Sometimes these pathogens, by binding to antibodies, they won't be destroyed by macrophages or NK cells. In some cases, like a toxin, basically the antibody binds around the toxin and it just gets excreted through urination or pooping. Ha! Real quick joke, not joke, but sorry. Reed was drawing pictures the other night. She drew me the poop emoji. Hilarious. Okay, sorry. And then she drew me Sloth and Chunk from the Goonies. So, anyway. Okay, so I think, if I'm not mistaken, I want to just see if there's anything else here that I have for us to cover. Okay, we'll come back to that. Let's take a look at this example on the right. And what I want you to do is actually to explain this one on your own. I'm going to give you a few minutes to kind of fight with it beforehand, but I think there's enough information there to explain what might be happening. There is a pathogenicity factor or a virulence factor hidden in this image. In other words, there is a protein produced by Staphylococcus that contributes to its pathogenicity. And it does so by inhibiting the immune system. What I want you to do is to look at this image here, and I'll zoom in if that will be helpful, but I want you to find what is that pathogenicity factor and then describe what's it actually doing and how does that inhibit the immune response. Okay? Give you a few minutes to fight with it. Oh, geez, I didn't mean to do that. Sorry.
is our pathogenicity factor? Kaylin?
Protein A.
Yeah. So protein A, what's it do?
It allows the immunoglobulin to bind onto it, which changes the shape, so it isn't able to bind to the phagocyte. So the phagocyte's not able to activate the phagocyte.
Yeah, exactly. And it's not so much that it changes the shape of the antibody so much, but it's as if these two are competing for the same thing. The Fc receptor and this protein A are both competing for this Fc region or this constant region on the antibody. And when Staph A does so, basically occludes or hides that region so that it can't bind to the Fc receptor. And so it avoids phagocytosis in this case. All right? So, Erica.
So, since like the variable regions on the not FC portion, do they still like have that and will be able to bind to that? Or is it just like chance?
Holy cow, that's a really good thought. So, not holy cow like I didn't expect that, but, well, I did not expect it, but not because of you who you are, but just because that's a really good thought. So what you're saying is that these could still bind? I don't see why not. In which case, you know what actually happens here is it's a process known as agglutination. We've actually used that term once or twice this semester. Anybody recall what agglutination or hemagglutination is? I hear, yeah, clumping. So you might, in this case, actually get clumping of the Staph aureus. I don't know if that's what happens or not in this case, but since these could still bind to those epitopes, you might get clumping with other Staph aureus cells as well. It's a really interesting thought. That could be a benefit, because clumps of pathogens are actually really hard to phagocytose. It could be a benefit for the pathogen. I don't know, but agglutination is a common way actually in which we inhibit the function of pathogens, particularly ones that have flagella. When they're agglutinated, they can't swim or move the way that they need to in order to cause disease. But Staph aureus doesn't have any flagella, so I don't know. It's a great question. I gotta go figure this out now. So, yeah,
My other question was, so like if something has an antibody on it,
yeah.
it binds to the pathogen, is it just, does it have to kind of just by chance float around and hope that it finds the macrophage with the, okay, so there's nothing attracting it?
No, there's not anything attracting it. And yeah, it is kind of by chance, but I think one of the things to remember here is throughout this whole process, there are cytokines being secreted by T helper cells. And not only do those cytokines not only are they involved in increasing the immune response, remember some of them are chemokines as well. What do chemokines do? They attract. And so yes, it's random chance, but there also is an attraction to that region to begin with, which can encourage this as well. The other way that this can that we get interaction between a phagocyte or a macrophage and these pathogens that have been opsonized, there's a couple. What system do we have? We have two systems in our body, one big one I guess, that is involved in filtering or localizing immune cells so that they can interact with each other. What gets swollen oftentimes when you get sick? A lymph node. The lymphatic system is designed, and we haven't talked about the lymphatic system, and maybe this is something we'll come back to kind of put this into context. But the lymphatic system, what we find is secreting chemokines too, pretty much all the time, that is bringing macrophages to those sites. These antibody-bound pathogens would eventually make it to the lymph nodes as well and can be destroyed there. The other spot, the spleen, is another location for blood-borne pathogens where we find high concentrations of macrophages and a filtering process of pathogen out of blood. Kaylin?
Is that why with mono, like that's where it gets inflamed?
Yeah, mono is a very interesting one because anybody know what mono and EBV infects? B cells. It actually triggers the B cells to proliferate. Where do B cells proliferate? In the lymph node.
And so that pain that you often get with an EBV infection or monoinfection is from the just sheer proliferation of those cells in the lymph nodes. Yeah. I can remember having mono, and that was awful. And it was during the summer, so. All right. Other questions on this? Good conversation. All right, last part that I think we should cover for today, and this kind of caps the process of innate immunity. The last big part of adaptive immunity. The last big part of adaptive immunity is the fact that it has memory. B cells and T cells, and we will not get into the details of how this memory occurs, but just state that it does exist. B cells and T cells, after they have been exposed to a pathogen for the first time, undergo a process of continual selection, where at the end of the infection, only those that are best suited to bind to the pathogen through its epitopes are the ones that end up surviving. And some of those cells become long-term, long-lived memory cells. There's a lot of debate right now as to whether, when we say that there's immunological memory, does this mean that we have cells that are maybe 30 to 40 years old that are just kind of lying around? Or does this mean that it's a pool of cells that maybe replicates at a really low frequency but kind of continues the lineage alive? And there's a lot of debate over which of those two is the true story right now. There's also a lot of debate as to where do these memory cells reside. We don't know the location where they are at, if there is a location. But in immunological memory, focus on the blue line here. What this is showing us is that B cells that produce immunoglobulin G, IgG, an antibody that is specific to the pathogen. First time you see a pathogen, you'll get this triggered response. And notice the adaptive immune response, we mentioned the innate immune response happens within like a couple of hours. Look when we start to detect antibody in infection. Typically 3-4 days. The adaptive immune response is fairly slow. Okay? But the first time being exposed to a pathogen, you get production of antibody to target that pathogen. And eventually when the disease or pathogen is cleared from the body, the titer or the concentration of those antibodies decrease over time. And then this is indicating a long period of time where there's no infection. The next time you're exposed to that same pathogen, not only do we get a much quicker response from the adaptive immune system because of these memory B cells or memory T cells that exist, but the response is much, much stronger. This is the basis for why, when we say if you have gotten measles, you will never get measles again. Or if you have gotten chicken pox, you will never get chicken pox again. It is this immunological memory that prevents second and third rounds of the same infection from occurring. Now, there are exceptions to this. There are some pathogens that seem to trigger a much stronger long-term immunity than others. And actually that's, a good way to think about this is vaccination. Vaccination, the goal of vaccination is to get you through this first part, expose you to an inactive or a weakened form of the pathogen so that you surmount some sort of immune response so that the next time you're exposed to it, you don't get sick. Some vaccines require what? Some vaccines require, if you get the shot, like tetanus for example, tetanus you have to get how often? 10 years, right? You need a booster to retrain the immune system. There are some pathogens and some antigens that are not as strong at triggering a long-term memory as others. Tetanus is on the shorter end. Okay? It seems like the HPV vaccine, on the other hand, is probably a very long-term one, at least from what we understand right now. So kind of keep this idea in the back of your mind, because this is something that we'll revisit in the last week as we talk about the process of vaccination, okay? I'll hand back exams, and again, the big things are, what I'm asking you to do is bring me a hard copy of your revised answers with the original exam by Tuesday. Staple it all together for me at this point. And you don't have to go back and re-answer every single question. You don't even need to rewrite the questions down. Just write down which number it is and write down your new answer. Okay? Hand-typed or handwritten or typed, I don't have a preference. Okay?
So all you need is the correct answer.
All I need is the new answer. Yep. This is now open book. And yeah. All right. That's not to say that all of these were bad. Actually, some people, this was your best exam, which you should be really proud of. Okay. So do take a look at it. Take some time to reflect on it. Emily.
Do you know like how much partial credit we're going to be back?
Yes. So what I've decided to do is that for the multiple choice or the fill in the blank, it's going to be a point for each of them. Okay. For the essays, I'll basically rescore the essay and then take the average of the first time and the second time to determine the new score. So, I mean, if you completely bombed this test, meaning if you got no points whatsoever, you have an opportunity to get 40 points back, roughly. Okay? Nobody's in that boat, so that should not be something to worry you. But I'm just telling you, there is some meat on the bone for everybody if you want to take it. If you don't, it's your choice. I'm not going to feel disappointed if you choose not to do it. For some people, they may just say, I'm fine where I'm at. I'm not going to worry about doing more of this. That's fine. Okay? All right. Otherwise, have a great weekend. Show up Monday. Donuts.
Question.
Yeah.
So I just found out about the